Bismillahirrahmanirrahim, and welcome back to this next video. And this is another video in the series of videos on the hemostasis. In my previous videos I talked about the primary hemostasis, and in this particular video I'll be talking about one of the types of the secondary hemostasis, which is known as the extrinsic pathway. So what is secondary hemostasis then? So secondary hemostasis, it actually refers to the cascade of enzymatic reactions, and these cascade of enzymatic reactions are carried out by the clotting factors. And when this cascade of enzymatic reaction take place, it ultimately results in the conversion of the fibrinogen to the fibrin monomers, or you can say the fibrinogen is converted into its active form, which are known as the fibrin monomers. Now what happens next is that these fibrin monomers, they are then cross-linked into insoluble strands that serve to stabilize the loose platelet plug or the platelet clot that is being formed in the primary hemostasis. So you can say that the secondary hemostasis is actually responsible for the stabilization of the loose platelet plug that is being formed in the process of the primary hemostasis, which is carried out by the platelets, and the secondary hemostasis that is carried out by these clotting factors. Now there are two types of the secondary hemostasis. One is known as the extrinsic pathway, which will be the focus of this particular video, and the second type, that is known as the intrinsic pathway, which I'll be talking about in the next video. Now when we talk about the extrinsic pathway is also known as the tissue factor pathway. It is called tissue factor pathway because it is initiated by the tissue factor, or it starts from the tissue factor. And it is also the shorter pathway of the secondary hemostasis. It is the shorter pathway because when you compare the number of the clotting factors that has been involved in the extrinsic pathway as compared to the intrinsic pathway, so the number of the clotting factor, that is less. Therefore this is a shorter pathway as compared to the second pathway of the secondary hemostasis, which is known as the intrinsic pathway. Now this extrinsic pathway, it is initiated by a tissue factor, and this tissue factor is actually produced by the endothelial cells of the vessels and by the leukocytes. Now when we talk about the tissue factor, sometimes the thromboplastin or the thrombokinase, it is actually used as a synonym for the tissue factor. So sometimes you would see in the literature that for the tissue factor you'd be seeing the synonym of the thromboplastin or the thrombokinase for the tissue factor, but these are different things. There are differences between the TPL and the tissue factor, because the TPL is actually a mixture of the phospholipids and tissue factor, which is found in the plasma. So the TPL is a more complex structure as compared to the tissue factor. Now this tissue factor is also known as the clotting factor 3, or it is also known as the coagulation factor 3. So let's talk about the pathway, how it happens. So when there is a damage to the blood vessel, what happens is that the tissue factor, which actually functions as a high affinity receptor for the factor 7, so the tissue factor is going to bind to the factor 7. And when the tissue factor it binds to factor 7, the factor 7, it moves from an inactive state into its active state. So when you see this small a with these clotting factors, that actually means that they are in the activated form. And when there is no a with this means that they are in their inactivated form. So this a is actually for the activation. This factor 7 is actually a serine protease, and I'll tell you in a while what are these serine proteases. So when the factor 7a it gets activated, what it do is it is going to act on the factor 10, which is in their inactive form, and when the factor 7a it acts on this factor 10, it is converted into its active form, which is actually the 10a. Now this factor 10, it is actually synthesized in the liver, and it requires the vitamin K for its synthesis. So just like the factor 7A, the factor 10A is also a serine protease. So the serine protease and the serine endopeptidase, they actually refers to like one and the same thing. Now when the factor 10A it gets activated, it combines with other things. So the factor 10A is going to combine with the platelet membrane phospholipid. This PL is actually referring to the platelet membrane phospholipid. It is going to combine with the factor 5A, and as you can see over here, this is in the activated form, and this factor 5 is actually activated by the thrombin, which is also known as the factor 2. So the factor 10A plus the phospholipid plus the activated factor 5 and the calcium, when they combine with each other, they are going to make a complex which is known as the prothrombinase complex. And this prothrombinase complex is actually working on the prothrombin, which is actually the inactivated form of the thrombin, and this prothrombin is also produced by the liver. And when the prothrombinase complex it acts on the prothrombin, it is converted into the activated form, which is actually the 2A, or it's also known as the thrombin. So this thrombin or the factor 2, that actually is one and the same thing. This thrombin is also a serine protease. So it's time to define what are serine proteases or what are serine endopeptidases. So the serine proteases, also known as the serine endopeptidases, they are actually the enzymes that cleave the peptide bonds in the proteins, and they are called serine proteases because the serine serve is the nucleophilic amino acid at the enzyme's active site. So we are talking about proteases, that means it is going to act on the proteins. So far the clotting factor that we discussed, the factor 7, the factor 10 or the factor 2, all of them, they are proteases. So they are proteases because they act on the proteins, and they are called serine proteases because serine is actually a main amino acid at the enzyme's active site. So when the thrombin 2A it is produced from its inactive form, this thrombin 2A is going to act on the fibrinogen. Again, it is produced by the liver, and the fibrinogen initially is in the inactive form. So the thrombin is going to convert the inactive form or this fibrinogen into its active form, which is actually the fibrin. And you can see over here, this is the activated form of the fibrinogen. The fibrinogen is also known as the factor 1, as you can see over here. So this one is the inactive factor 1, and this one is the active factor 1. What happens is when the fibrin or when the factor, it's factor 1, it moves into its active state, it is going to combine with the factor 13, or you can see the fibrin is going to act on the factor 13, and when the fibrin acts on the factor 13, the factor 13 is going to move its into its active state, which is the factor 13A. And this factor 13A is actually a fibrin stabilizing factor. So the factor 13A, when it combines with the calcium, it is going to stabilize the fibrin plugs that has been formed by the fibrin monomers. And as you can see over here, this one, when the fibrin monomers, they combine with each other to give you that polymeric structure. So the factor 13 is going to cross-link them, and when they are cross-linked with each other, that means that the fibrin plug that is going to get stabilized, and when that gets stabilized, that is going to stop bleeding from the damaged blood vessel. So this is all about the extrinsic pathway. So if you like the video, please subscribe to my channel, hit the like button and share it with your friends. And in the next video, I'll be talking about the intrinsic pathway, which is the other type of the secondary hemostasis.